Come and sit down. Come and get involved in this. What's your name? Come and have a seat. What are you doing here? 10 minutes is the perfect amount of time. You'll be surprised how far you get in 10 minutes on Talkaoke. The whole point of Talkaoke is to talk about whatever you want to talk about. It's kind of up to you. So what's your name?
So,
And what was it again?
Hashem.
Hashem. And my name's Mikey. What are you doing here, Hashem? What's your interest?
I'm interested in data analysis for genomics.
Oh yeah.
Yes.
Yeah.
So I'm a PhD candidate in statistics.
Okay.
Yeah.
And have you found anything here that's kind of...
Yeah, there's some pretty exciting technology going on here. So if you've heard of Oxford nanopore technology.
Yes. We've got this person. Come and have a seat. Well, it's kind of up to you. It's you're the perfect person then for Talkaoke. Come and sit down and listen. What's the Google? We can put out a call for Google Brain. Where are you? There we go. We have got Stephen is looking for you. Come and sit down for a second. We'll tell you. We've got Hashem. We've got Saul. Okay, laters. We'll see you later. Anyway, you're talking about data analysis. You found some interesting. You're talking about Oxford nanopore. Do you know what Oxford nanopore is?
So are you familiar with gene sequencing? So the ability to read genetic information. So right now, how it happens is very similar to how when the computers started first. A computer used to be a whole building, right? So you can't really walk around with a laptop. But then they made it smaller and smaller, and that's what nanopore technology does now. It's something this big. It can plug into your phone actually, and it sequences any sample DNA. So you can actually go anywhere with it.
In your hand sequencing technology. Yeah, I heard about that. I didn't know what it's called.
Yeah, they have it in like the space station.
Yeah.
They have it in. They took it to Antarctica, the desert. You can take it to West Africa and sequence like malaria, Zika virus, and all those things.
And why is it that exciting for you as a data analyst?
Well, for me, it means that I can get more data, basically from all these different locations. I can just have it in my pocket and just walk around with it, grab data from any biological samples, and then just put it in my laptop and crunch numbers. It's very exciting. It's exciting times.
So you're actively searching for more data from diverse sources.
But not any kind of data, genomic data.
Yeah.
But especially, so they're still working on getting the accuracy.
Yeah.
But they're doing a lot of great work.
Yeah.
But yeah, so if it's more accurate than like the leading technologies like Illumina or Agilent, then yeah, they're basically gonna dominate the market.
Where do you see this technology going? Come and have a seat. Give us a couple of minutes because we've got Hashem, we've got Saul. What's your name?
Anelia.
Anelia. At the moment we're talking about the Oxford nanopore technology. We're talking about the fact that you've got this much. When it comes to computers, you've got this much power in what used to take a whole building, and it's the same in a way with genetic technology, that you've got this much power now in a tiny little device. And Hashem's very excited about that. I don't feel that Saul is that excited about it.
Because I don't really understand how.
Yeah.
If it's just more data, it is a step change in the technology, and I just don't understand enough about it.
Yeah. Are you excited about this?
About the nanopore?
Yeah.
Actually, I have a little device and I have the mini ion.
Yeah.
And I've tried it for RNA sequencing.
Yeah.
So one of the things that happened was that the protocol took much longer than we expected.
Yeah.
And then my laptop that was dedicated to that broke, so we couldn't continue. So I cannot say much.
Yeah.
But we're gonna try it again.
Is it like a shiny new toy and you've got a lot of expectations for it?
Actually, you do. I mean, we did. We got a lot of expectations.
Yeah.
So one of the things that is kind of I mean, the low throughput of it was something that we needed to play around to actually get it to work for what we were imagining it's going to work.
What were you imagining it was going to work for?
I mean, we were hoping to get rid of abundant transcripts and then to get kind of a representation of highly abundant transcripts.
Right.
But like the whole idea was to be very quick.
Yeah. Quick
And because we couldn't make it very quick, this is when we actually kind of sat down.
Sorry, we're not actually talking about that. We're talking about something completely different. We are talking about, well, in a way it could relate. We're talking about the Oxford nanopore technology, you know, something as big as this to get genomic data. Is it slightly overhyped? Is it going to lead to a whole new world? Saul's not very excited about it. Hashem is very excited about it. Analia has tried it, and it's still, you know, a bit kind of shonky in terms of what.
I have a question.
Come and sit down. Come and have a seat. So we've got Hashem, we've got Saul, we've got Analia. What's your name?
I'm Stefan.
Stefan. Do you know anything about this? So you've got a question. What's your question?
Yes, so because he's doing single cell sequencing, I was just wondering if you have tried
Have you tried this?
No, we haven't tried it. I heard many things about it. Many people are very excited about it. But I also heard that sort of the error rate is a problem.
Yeah.
Right, and that you have to throw away a lot of your data essentially.
Yeah.
And so I'm sort of, more like wait and see where this is going, and maybe in a couple of years when sort of these things are.
Yeah. Do you think it's like more like the iPhone? You know, you wanted to wait for the next generation, really.
Yeah, that's how it feels. I think in general it's cool that it has such a small form factor, right? And you can just I guess the smallest thing, you just hook it up to your laptop, right? By USB. And that's kind of cool, you know, that everybody can.
Yeah.
By the end of 2017, you'll be able to plug it into a phone.
Right. So you won't even need a laptop. You just go with will it do Android and iPhone?
They didn't really disclose that much information.
Okay.
But there was a talk at 2:30. That's where they were.
I mean, why can you just explain a bit more why you're excited about it?
So I'm excited because I want to work on. So for me, I see the fact that the error rate is high as an opportunity because I work on algorithms to control the error rate.
Right.
Yeah,
so you think you can actually
make
iron out
the error rate better?
Yeah. I mean, can you say without kind of blinding us with science how you're going to do that? Can you explain it?
I can't, like, a priori, you don't really know if it's going to work until you, like, test it and compare it to their models. But right now, I believe they're using neural networks to do that kind of base calling, they call it.
Right.
So there are other approaches.
Yeah.
You can do other more statistically rigorous approaches because a neural network, you don't really know what's going on inside. It's just like a
black box, really.
Yeah, you don't really know what's going on in the hidden layers and stuff like that.
Yeah.
Interpret why is it calling the bases in the wrong way and stuff like that. Maybe having a more principled statistical approach would make it maybe understand.
What do other people think? What do they think about the neural network? Or
I was just wondering, this error rate, doesn't it come from the very raw data signal, like how you change the voltage, how well you...
So they're using a neural network to translate the potential signal into bases, ATCG. Yeah. So is that...
Are you saying that it's not precisely enough how you determine the differences between how the different bases change this?
Yeah. Okay.
Right. So that's the thing. And you believe that you can improve that by...
Yeah.
That is one avenue. That is one avenue you could go about improving.
I mean, what about this question about kind of artificial intelligence doing the science and you don't know how it's doing it? I mean, how do people feel? I guess you're scientists around the table. How do you feel about that approach to science where you've kind of got these funny little machines recognizing these patterns or whatever they're doing?
I actually feel pretty good because I believe that there are patterns that we may not be able to comprehend just because they're like so complex or contain multiple elements that not all of them we still understand. And for a machine, it's much easier to drag, to kind of infer patterns and stuff like that. So I do believe in that in general.
just too much data, right? There's just too much data to go through it all by yourself, right?
Yeah.
So I think machine learning is a very well accepted and great tool, right?
Yeah.
If I would call it artificial intelligence, I'm not so sure.
Yeah.
I think.
You don't see it that way. Yeah.
Exactly. So it's statistical learning. It's you know, and it's a great tool. But I think I would call it artificial intelligence if the computer can come up with hypotheses and invent experiments and write the paper. Then you know we're obsolete.
Yeah, are you worried about that?
Right, especially write the paper.
Yeah.
Okay.
I mean,
I'm not worried about it. You know, I don't think in my lifetime. Maybe I'm too pessimistic.
Yeah.
But I don't think we'll be that easily replaced.
Yeah.
That's awesome.
Yeah.
And another question for you. So you mentioned today, I listened to your talk when you said that when you're dealing with single-cell sequencing, one of the biggest problems is the lacking data, like the inability to recover the major part of the RNA, right? You said on a good day at 10%.
That's right.
Yeah.
How do you deal with that?
Well,
Computationally.
Computationally.
I mean, not only computationally, but I would imagine you have also computational approaches. I guess biologically you deal with multiple repeats and stuff like that.
Right.
But is there a computational approach there?
I don't think so. I think this is just a basic limitation of the method, and you just have to understand sort of the type of questions you can potentially answer, right? So, for example, if your favorite gene is just very lowly expressed, then you know this method will not tell you much about it. But you know it is great at just classifying different cell types, stuff like that. But I don't think there is necessarily a computational way to just make this issue go away. It's rather, it does present a challenge for analysis because sort of the statistics of these dropouts, right? So that's there's a lot of zeros in the data. That is something new, and we have to try to understand how to deal with it. But sort of this fundamental issue is not going. You know, you have to improve the method itself, the wet lab part of it, you know, to improve that.
Question then, having this low recovery rate, do you believe is reliable to use single-cell sequencing for allele-specific expression?
People have, I mean, that has been published, right? And I looked a little bit into the data, and it looked pretty good to me. I guess you want to use relatively long reads, right? So you can, ideally, you capture multiple seeds in your.
Even if it is one SNP, I was just imagining that if you lose the signal randomly to a very high rate, you can get like wrong estimation of the allele prevalence. Let's say for a heterozygous SNP, where you want to see which of the alleles is expressed at the level of RNA, then if you get such a high error or low recovery rate, you may recover.
I just want to make sure, are you following this or Hashim?
I am, but as I said before, I have a talk in it with 3:30, but it was really great talk.
Give us a final thought. I mean, what you know, Stefan, I want a final thought from you as well before you go. Right. Give us a final thought where this conversation is going to go. Let's get a final thought from you first of all, Hashim.
Well, as I said, a lot of people tout the technology as being very. It's going to open up a lot of avenues, especially for its low cost. Experiment, like you can do a lot of experiments with it, especially in underdeveloped countries, like Africa, West Africa. I'm from North Africa, so especially for me, I can take this back home. And, you know.
I mean, earlier on, actually, we were talking about this idea that one way to sort of get a wider sort of understanding of the culture of genomics is to make the technology more accessible, more available, and get this almost kind of like garage, you know, experimentation going on all around the world. I mean, do you see this as a kind of way to make that happen, or do you think it's a good idea to a more kind of low down and dirty approach to genomics?
Well, I think they would be more. I'm not sure. I think it's early to say at this point. Because I think Oxford Nanopore is the only company that actually produces
something like that.
So I guess there's still a bit of time before we can actually see it.
But in a North African context,
nanopore technology is there, so
it's
just a matter of perfecting it more. And I see it as an opportunity as someone who makes machine learning algorithms.
Something to play with and actually.
I mean, People are applying it to make your phone order pizza for you.
Yeah.
So applying it on something that has potential impact, like calibrating nanopore technology or stuff like that, is for me is much more worth it.
Yeah. Not that there's anything wrong with pizza, unless you're a gluten-free type person. Stefan, have you got a final thought?
About the nanopore?
Well, just anything really. I mean, you know, we're going to be carrying on this conversation. Where would it be interesting to take it?
Okay, so you mean in general genomics?
Yeah,
or,
I mean, you know, I've got people here. They might be, you know, they could be anything. They could be geneticists. They could be bioinformatics. They could be students. They could be patients with a particular kind of genetic disorder. You know, what are the big subjects do you think?
The big subjects. I mean, so I'm very much interested in single cell heterogeneity, right?
Yeah.
And that field has exploded over the past years, especially single cell RNA-seq. So I was in a session earlier. Half of the people are already doing it, and I think that's great. Sort of that, you know, that's become so quickly so pervasive. And I think a lot of interesting stuff is going to come out there. Yeah. But a little bit, you know, now it's a little bit, the excitement is, you know, very big. But we still have to think about really clearly what are the interesting questions scientifically that we can answer. Like sort of the methods and technology is sort of racing ahead, you know, and there are many possibilities, but my feeling is we still haven't figured quite out yet what are the best questions to ask using that technology.
Just throw in one question. What would be the one question?
Well, I mean, my lab is very interested in developmental biology, right? Yeah. So that's sort of the fundamental question: how does a complete organism develop from an individual cell? Yeah. Right. So that is the big picture question. And I think, you know, the single cell omics will help us a lot answering that question.
Yeah. Okay. And I mean, I could go a lot further, but you know, that was supposed to be a final thought, not an initial thought. But you know, that's definitely food for thought. Thank you, Hashim. Where are we going to go with this conversation? Have you got something, Saul?
Not at this time, but I'm excited about joining the conversation without my laptop in front of me.
Okay, well that's good. Well, you know, everyone these days, we've got a lot of social media going on. I heard quite a few pings and dings over from this side of the table. But, you know, this is the world of constant communication that we live in. Why can't, you know, genomic data be part of that part of that continual toing or throwing of information across the web between people? And Elia, where do you want to go with this conversation? Well, I mean, you know, we're here, it's 3:30, you know, there's a few people standing around, we're at the Festival of Genomics, it's just you, me and Saul. At the moment, we're hoping to get some more. Maybe this man's going to come and join. Come and have a seat. 2 minutes. Give us 2 minutes. It's you, me, Saul, and what's your name?
Yogeshwar.
Say that again.
Yogeshwar.
Yogeshwar. Can I put Yogi's okay? Okay. All right. I'm Mikey. We've got Saul. We've got Anelia. We were just kind of at a point where what do we actually talk about? We're here. You've got a couple of minutes. You know, we want to get the juice out of you, obviously, but you know, people have got coming from different points of view. But we never really found out what people do and why they're here. But what is the conversation we should be having that isn't being had? Yogi?
Well, I'm coming from an academic background to industry just the last 2 weeks.
Okay.
I was studying insect genomics before I came here, so this for me is introduction to human genomics and where medical field is right now. Yeah. So to me, this is very good. Yeah, maybe, and the range of talks and the topics covered here is pretty broad. Very good introduction.
Do you feel like you've kind of arrived? Is it more interesting than insects? Did you say insects?
Insects, humans, bacteria. I was doing everything. I'm an evolutionary biologist. So to me, system development. Right now, I'm doing immunology, which is part of biology.
Did you say that you just moved from academia to industry in the last 3 weeks? Wow. How's it going?
Still in the shock, but I'll get there.
So you're having is it a culture shock to move from one to the other? Is it? I mean,
well, if you are in the area of a broad institute or other or medical university or medical schools, there is not probably so much culture shock. So I had collaborations with medical schools before, and I felt like the environment there is very similar to what's happening. So the convergence between med schools and industry happening. If you're from the science side, then yes, there's a shot.
Okay.
I'm curious about. Are you in the academic field or you're an academic?
Are you an academic?
I am.
Yeah. Okay. So we've got two academics, one former academic,
and one escapee.
Yeah. Yeah. I mean, have you ever? You're an academic. Have you been tempted to go across to the other side?
No, I don't think it would work for me. I just don't think I'm into that kind of career. So no, I didn't think about it.
Okay. Are you going to try to tempt, or are you going to happy to let us stay on the other side?
No, I was like that in January. So. I mean, I just.
What tempted you across to go into industry?
Well, one is my CV got picked up. I was called for an interview. I just decided to give it a try.
So you got persuaded in a way.
Well, a friend passed around my CV.
Okay.
I was tempted to try. I came here and started to one of the companies where I didn't go. I was, it opened my eyes to what the kind of research is happening in research institutions within the industry.
Okay.
So that kind of changed my outlook in general.
I mean, is it easier in a way to do research?
I don't know.
You don't know yet.
I'm still going through. I'm still going through orientation. So I'm sure it's different. It cannot be easy anymore, but it's different.
Yeah. But you're on the academic side. I mean, what have you got a view on this? What do you think?
It's extremely challenging. It's very difficult, and very few people kind of, are able to succeed to the level they want. That's my impression. It's a lot of struggle, very low paid, very talented people, which is unfair, I think, also. Because many people actually go through a painful postdoc, and then there are not enough open academic positions for them to grow. So that's a big challenge, really.
So it's a kind of
and if these people have decided from the beginning that they would go to the industry, that's probably more straightforward path. But it's like it's imbalanced in the way how many people are actually interested and capable of doing it, and how very few positions are offering for more advanced stages of the career.
Okay, so in the sense we're hearing how it's such an expanding field, but in terms of academia, that's not expanding to meet at least the career needs of the people out there that are fit and ready and able and willing to do the research, keen to do the research, but there's not the academic pace for them to do it.
What's going to get younger people involved as well? That's the other question, is if there's not a career path within academia, well, I guess people might just use it as a stepping stone to get into bioinformatics. I mean, I know because there were so many people doing bioinformatics in my PhD program, and they all knew that they were going to get jobs immediately afterwards. Not a single one of them wanted an academic career. And indeed, they all got extremely good jobs immediately after they graduated.
Okay,
I think even at med schools and universities, they are training students to keep their minds open from the very beginning. Unlike when I was doing PhD. So things are changing pretty fast.
I mean, do you think there could be more research posts out there, though? Considering, I've been hearing all day about this kind of expanding field and a lot of hype about the possible research that could be done. I mean, are we creating enough posts for this to actually happen, or is it being kind of narrowly funneled into, a few institutions?
The institutions that are funded, I'm sorry.
That are funded. I mean, because it is in a way, it's what we're really talking about is
it's a circle,
money.
actually. The money always goes to the people who have the most money. So that's what happens in the research too. Unfortunately.
Okay, so the money follows the money. Pretty much.
But I think that you could have, if, for example, the technology changes, then you could have people doing this outside of institutional context. That's, I think, probably one. If you look at the way that the software industry worked 20 or 30 years ago, it's really changed in that respect, because suddenly you can do the sorts of things that would have required huge mainframes in your bedroom or in your iPhone. So maybe the same could happen with these smaller, more convenient technology.
It could be kind of almost like a home research. Just get a final thought from Anelia.
on?
Well, just whatever you're thinking. Just, what are you going away thinking?
Okay, so I'll just say something that passed through my mind when you mentioned that.
Yeah.
No, actually, you mentioned bioinformaticians.
Yeah.
I have a feeling that there will be a boom for like a high demand for bioinformaticians, which is going to go down. Is my feeling again, just because yes. Because
so the machines will take over.
Yeah, so I think it's a moment now, and actually it's very exciting to be in this field. But then setting up of the analysis doesn't take long as a real active working time. So that I don't know. That this is also disappointing.
I really noticed that actually the people who were doing the glamorous research, i.e. the research that would attract the funding for longer term investments, for lab setups, were the biologists, not the bioinformaticians. And the bioinformaticians were like Scotty on the Enterprise. It was not a glamorous role. It was a required role, but it wasn't a glamorous role.
I mean, have they got a very short shelf life, the bioinformaticians? Are they all going to be an extinct species in the future when the machines take over? Yogi, you look like you're shaping up to leave. Give us a final thought.
Well, I'm a biologist who uses computers, so I have a different way of looking at things.
Yeah.
So I think there'll always be role for biologists. Doesn't matter what tools you're using.
Yeah. Okay. So you feel safe and secure and confident, and the world is your oyster. And good luck with the transition.
This is a talk show. It's Talkaoke. You can come sit down. I can't hear you from over there. That's fine. Well, that's the point. We have like a broad area about genomics, but we don't have to stick to the script. It's actually up to whoever sits down around the table.
At the moment, we're talking about can you have science without bosses?
That's the topic of conversation. No, not at all.
No. In fact, it qualifies you.
So I'll just explain quickly. This is Talkaoke. It's a kind of talk show. So far, the people we have around the table are Mikey, and what's your name?
Paul Ventura.
Welcome, Paul. Can I call you Paul? Okay, great. My name's Saul. I'm hosting this, which just means I'm moving the microphone around. So what we've been talking about, I mean, the biggest question around the table today has been, should you edit your baby? Which I think is maybe a bit of a red herring. Everybody goes to that question immediately, but maybe I'm wrong. The topic on the board at the moment is genomics in the media, but I don't think that that's been a very popular topic. So it's really down to the people around the table to decide where we take the conversation, and I'm curious if you've had any experiences here yet today that have given you any inspiration, food for thought. Have you been to any interesting talks?
I walked in 5 minutes ago. So
Okay.
I felt a little unqualified.
No, well.
But I certainly would not edit the baby.
Okay, so that you already know your position on that, so maybe we can move on a little bit. I mean, is your interest here, I know it's not done always to ask somebody why you're here, what you're doing here, you can do whatever you want here, you don't have to tell me, but you said you're not a scientist, what is your interest?
Yep, we just launched a new digital PCR platform, so we're here to show what our new technology is capable of doing, get some interest gathered, hopefully find some new beta partners for us to continue our research and hit the ground running. So.
Okay. Fantastic. You're not actually the first person who's sat down around the table who's just doing exactly that. That seems to be a big part of the industry at the moment.
It's right here in Boston. We're just outside of Boston, so this is an excellent opportunity to bring in talent. And locally for us, it's an economical show that allows us to get access to top researchers that can see what we're working on, and hopefully begin to think about us as we commercialize our efforts.
Okay. Yeah. Go ahead.
Because I'm not from the area, why is there so many companies like this in Boston? Is there something about Boston that attracts this kind of technology?
I think Paul might be the person to answer
Sure.
that.
So you have the MIT, Harvard, obviously that's a big gene pool of talent that comes out.
No pun intended.
From that you've got, of course, the Broad Institute, which is an affiliation between Harvard and MIT, so you get a lot of postdocs that go there for a couple of years stint, come out, genuinely like the area. A tremendous amount of software development that's happened in the economy over the last 20 years. So the infrastructure is solid. It's heavily VC and private equity backed. So that, on top of all the research hospitals that are here, it lends itself to be one of the top research spots in the world. I've been here 25 years, so I know my job's secure being in this industry, and this is my backyard.
So is the big problem recruitment? Is that the major bottleneck? Is it finding people? You're describing this as a sort of talent pool, gene pool. Is that the biggest problem for startups in this field, is to find the people to do, and what is it? You're looking for researchers, biologists, bioinformaticians?
We look for robotic engineers. So we have had a nice pool of talent, because if you're coming out of a top educational facility in this area and you want to stay in the robotics field, there's only so many players in town for that. There's also the people that like startup mentalities, where they're willing to take more of a risk, have a bigger equity stake in a company that may or may not fan out. But then you've got also the backbone in town, which is the Novartises, the Pfizers, the more Mercks, the stable companies that can employ you for decades. So I think Boston gives you the bit of both on that side, either the adventurous one that wants to really get into cutting edge technology, or the one that just wants to just do their research and know that they hopefully have a job in 10 years time.
So is there a future in the robotics field? Is the idea that we're going to have artificial, not necessarily intelligences, but at least replicating systems that are able to design new systems? Is that a risk for the people who you fish for in this neighborhood? Or do you think that's
no,
the
it complements
future?
everything. So we sell instrumentation that miniaturizes assays, makes them more automation friendly, and not in one instance can I say it has eliminated a position.
Right.
If anything, it's created a need for someone to be able to do more work, do data analysis. It kind of complements their position.
Right. Okay. Well, yeah.
Because I mean, a bit earlier on we were talking about is bioinformatics in a way going to be like a shrinking pool once the computers take over and they just do this kind of like incomprehensible number crunching and pattern recognition, and actually we kind of human beings can't actually even keep up with it, and it's the actual algorithms are generating themselves.
Well, this was a senior, just to recap a little bit, this was a senior scientist who is an academic and is in the academic field, and was saying that essentially there's a boom in the recruitment of bioinformaticians, but that setting up the kinds of studies that actually are about biology, that's what the scientific inquiry is about. That the informatics part of it is only a kind of mechanical process that could be automated in the future. But I think you may be talking about a slightly different field if you're talking about miniaturization and about robotics.
Yeah, exactly.
Yeah.
But you can't, I look on the job boards frequently as we're trying to recruit, and there's not one job posting I don't see that doesn't have or a company that doesn't have a bioinformatics interest.
Yeah.
So if I could go back to my college degree, I may have switched up.
So do you think, you mean you would have gone in for bioinformatics?
They're highly sought after positions. A good talented writer, software developer can command a very heavy pay with a lot of options.
Yeah.
And I think that's a fairly safe industry to be in at the moment.
So the other thing that we had around the table was one academic who just left sort of 3 weeks ago. He just moved from academia into industry. And it must be an issue for you if you're recruiting people out of academia, which is a very strange, in a way, it's a very strange employment field. One of the senior scientists was saying there's no senior positions, there's this kind of bottleneck at the postdoc level where people have to leave because there just aren't senior scientist positions. Do you see the friction as people move from academia into industry? Because you must be the one that ushers them through that process now.
Our experience has been we found better luck with coming out of college, coming out or coming out of a PhD program.
Okay.
And the philosophy of our owner has always been that they've never been told no before. So knowing that you give someone a problem, if they don't have the experience to know that it can't be solved, then they go at it with their own intelligence, their own plan on how to solve it.
Okay, so
they fail, but sometimes they succeed when someone else would have maybe had the thought to think that this program wouldn't exist or couldn't be solved.
Right.
But these younger people that come through haven't been told no.
So ambitious naivety, that's what you mean.
What about this question about, because it's such a competitive field, that are you worried about losing people as well? That people just move on and they get poached by other... is that something, do you just see it as a kind of just a progression and that's just part of the field, or are you sort of like, oh no, damn, we lost another one?
It's a good question. We lose people because we're getting them so young into their careers. Either they find a different location in the country or world where they'd rather relocate to. They don't know any better, so then they see a friend that's working at a job at a different company that's like, oh, you have happy hours on Friday? We don't have that. I like that idea. Maybe options become something that lure people away. But we found in the engineering side of things, if the work is good, we get good talent, and as long as they're excited about the projects themselves. I head up sales, so I have more turnover in a sales environment where people can get recruited and poached by a new technology or a section of the company. That's fairly routine, but the engineering side we've been able to retain just because the concepts and the technology we're doing is interesting to them to keep them, but that's one side of the business.
That's interesting. So it's actually the quality of the ideas or the quality of the work, the challenge that it represents that will keep engineers around, but salespeople, they're just interested in something that's sexy, or is it they who are interested in the happy hour on Fridays? Okay, yeah, go on.
I mean, in a way, it's a part of the kind of, the sunken cost fallacy that, well, it's not a fallacy, it's just a thing. If you put in work, effort, mental and emotional effort into something, and I imagine as an engineer, you put a lot more into it, then you feel more invested in that. So you're going to stay for longer, aren't you? Oops. That's, don't know what that is.
Okay, I think that's actually something that keeps academics working away as they do for low pay and little expectation of financial reward. They're very invested in the ideas, and I wonder whether that gives them. I mean, it sounds to me, if you're recruiting people straight out of college, maybe it's a disadvantage to have people who are just like maniacally keen on a particular idea or fixed in a particular way of doing things, and actually that kind of youthful naivety is worth capturing. But recruitment, I mean, this has been another theme around the table earlier on, people were talking about the fact that there isn't really the high school and college level education that's going to meet the needs of this industry in 5 years' time. It seems to be exploding. Are we going to find ourselves in a place where there's just not enough people who are excited about it, who have that commitment to it? I mean,
open-ended, right?
Yeah. Go ahead.
Give us a final thought before you go. What's your name again?
Paul. Yeah, Paul, do you have any final thoughts for the table just about generally genomics or your interest in it, or what would be your sort of message for the future of the field if you were going to give a message to the people who might be thinking about going to college, studying these kinds of things, you might want to recruit?
So I've been in the industry for 20 years, so I've seen human genome project accomplished at an extraordinary cost and didn't believe any of the models that were out there that said it would, human genome would be done for under $1,000. That seems like a pipe dream just even 10 years ago. So I'd like to see where it's headed next because the cost factor has been eliminated. That's why we're here today. There's more companies spawning up. Things are becoming detected at a much lower detection level. And it's exciting. There's no other industry, I think, that is nearly as sexy, is nearly as well funded. And if you've got a good idea, there's money out there and people will invest in it. Gene editing is coming along to the point where anything can be made, it appears. There's risk there, of course, but it can be done and it's just going to continue to improve. So I'm anxious to see how legislation begins to get involved in all of this and regulation, which unfortunately will spawn sooner or later. But right now, let's see where it goes. So we sell equipment that helps detect some of that. So for my own personal gain, I see us as being in a good position for this market, and I'm excited to see where it goes.
Are you worried about regulation? You sort of seem like
we're R&D based, so but it just seems like it's inevitable.
Yeah.
It's a golden box that anybody can take and either do powerful, wonderful things with it, or equally it could turn into something that you wish you never opened. So that's to be determined. But I think without any regulation, it could get ugly. I don't know. It's the unknown, right?
Right.
I mean, just a bit earlier on we were talking about this, and the consensus around the table seemed to be that we can't regulate yet because we don't know what we're doing. And I guess that at that point you've just got to, it's coming later, but not quite yet.
Yeah. Okay. Well, great. Thank you very much, Paul. Nice to meet you. Okay. It's just you and me, Mike.
Again, it's just like the old days. Just like, I don't know. I think you're gonna, I think it's sort of dying down anyway. Well, maybe we should get like a final thought for ourselves. At least.
There might be a few final thoughts.
Yeah, we could even possibly just farm a couple out. And, sorry, Rick, you'll probably have to edit out the sort of long pauses in between. But we are.
Come sit down. Come get involved in this. This is kind of talk show. You're almost here already. Okay then. What's your final thought?
I would say that in a way the best way to get people to sit down is to just assume that they will sit down and just start to you know, just say what's your thought? What's your final thought for today? Or something. I don't know. It's a difficult one. But I guess when it coming back to the discussion, I think compared to last year, I think the hype.
Come sit down. Come get involved in this. We're just wrapping up for the day.
So 2 minutes. 2 minutes.
Well,
2 minutes.
We're trying to hear thoughts from all over.
We need closure. We need some kind of conclusion, some kind of final, final sort of wrap up.
I can't hear you from over there. Just come sit down and share some thoughts with us about last night. No?
No.
Just come give us your thoughts about the day. We're just wrapping up here.
Basically, saying you're not attractive enough. So
Okay. Right.
that's the. But maybe, maybe FLG, the frontline genomics, the organisers of the. Yeah. Maybe we can ask Lara, I suppose, if she wants to have a go in the middle. I've got a feeling that the answer will be no.
Yeah. At this stage.
Yeah.
I think that's right.
But there's no harm to ask. But yeah, I guess it's the final wrap. I think, as I was saying, I think there's a lot less hype this time around than there was last time we were here in Boston, and certainly, I think, as actually the reality comes into play, the hype in a way goes down. But
That actually,
that leaves us with less space to kind of imagine and be excited about stuff as well.
Do you think this industry on the way out? This industry is now sort of fading?
It's not on the way out, but the thing about what you call bleeding edge technology is that quite often when it's easy to do, people are less interested in doing it. It's like the thing with streaming. I don't know if you remember us struggling with like 3 computers with Real Player and a postage size kind of collection of pixels.
Yeah.
And trying to configure the server, and then everyone was excited about streaming. Now I could just have streamed this on my phone, but nobody gives a shit. Do you know what I mean? The same number of viewers, but it's just like when it becomes easy. When it becomes easy.
Facebook streaming has changed that a bit. It's become easier. There are more viewers and more people doing it. It's not true.
But I mean, we're not doing it. Put it that way.
We were never interested in
and it's the same with the bleeding edge technology. Like as it becomes more possible, then probably the hype goes down.
Do you think that you could apply? Okay, here's a question for you. Do you think that if the technologies that are available to us for communication, such as the Talkaoke table, are available to us for gene editing, that it would be possible to do a project that deals with that uses gene editing to do the same kind of open-ended kind of sharing of information, not obviously talking about information conveyed through language and social interaction, but information conveyed through genetics. And what would that look like? You can imagine a people speak project that involves genetics.
I mean, if we can work it out, then the funding is there. Do you know what I mean? If we can work it out, if we can do that, then the funding is potentially immense.
This is a good place to do it because we've got plenty of time to talk.
The funding is potentially immense from that, but I guess it would be, and it goes back to this non-hierarchical science question. I'm sorry to say, but
Come sit down. Come get involved. This is Talkaoke. It's the kind of talk show. We're talking about whether you can have non-hierarchical scientific ideas.
Can you have science without a boss?
Can you do science without a lab PI? Come sit down. What's your name? At the moment, it's the people around the table. This is decorative. So far we've got around the table. We've got Mikey. What's your name?
Abby.
Welcome, Abby.
Julia.
Welcome, Julia. My name's Saul. I'm facilitating this talk show at the moment. So what we have been talking about is whether or not it's possible to have scientific ideas without having bosses. Can you have a non-hierarchical form of scientific practice? That's the question. We don't have to talk about that. This is Talkaoke. You seem to have an opinion.
Only once you've accomplished your foundational space.
Now that is an interesting concept that I have never heard about before. Do you know what foundational space is? Do you know, Abby?
I kind of know what she's talking about, but I don't know.
It sounds like you're going to have to explain this for everyone.
So I mean, like, you know, basic college level science, chemistry, biology, physics, math. Hopefully some writing in there too would be nice.
Right.
I think art promotes science well, so I don't think you can be a scientist without also being an artist.
That's unusual, but a wonderful opinion.
You know, all of the original scientists were absolutely artists, even had to draw what they saw in the microscope.
Okay.
So it's essential that they were able to actually do faithful art.
Okay, that's interesting. So sort of observational scientific methods depended on those kinds of abilities. So how does that, wait, just before, I see you've got something to say about that, Mikey, but how does that lead us? I love the idea of this universal education in all these key fields. How does that lead us towards the idea of non-hierarchical science, if that is the direction it's leading us?
So I think once you've been schooled in those foundations and you have a sense of what the scientific method can actually accomplish with, you know, hypothesis generation and testing, then you have a framework with which anyone can engage.
Okay, so it's the sort of basic idea that rationality isn't about hierarchy, it's just about being critical, being human. Everyone could do it if they have the tools. Mikey?
I mean, you know, in the sort of era when science was actually establishing itself as a force, and you know, the Victorian scientists going into the early 20th century, there was no institution. There were institutions like the Royal Institution, but there were no big laboratories. It was just individuals doing science, and I think it worked well.
Is that the future? If it was the past, is that also the future? Is that what you would like to see, Julia?
No.
Oh.
Because that relied on the aristocracy.
Okay.
And so, how can you accomplish that and still have a sort of middle class approach? Do you have to be an independent entrepreneur or inventor? Or can you do that within some kind of framework? Can you be a citizen scientist?
Okay, well that's a very, very complicated and interesting question. Abby, do you have any, what do you think about this idea? I'm aware that you said you sort of know what Julia was talking about. Is this what you thought that she meant?
Yes. So I still think I try to compare, you know, even if you know exactly what the science is method is and everything, you need someone who can pull everything together. So I think the communication is missing. So in that, is that the boss you're saying, maybe able to bring everything together, divide or figure out what needs to be done.
Okay, but does that have to be the boss? That's a slightly different question.
Yeah, I mean, in
go on.
In a way, it's based on a consensus. Science is based on a consensus. So you don't need a boss for a consensus. You just need enough people to agree with you.
Is that kind of what you think? Consensus is possible?
So I was going to say experience, right? So you could say instead of boss, you could say experience. So you need some mentorship, or you need your endeavor to be, tiered in some knowledge of what's come before you.
Right, so it's less about hierarchy and it's more about knowledge or wisdom and experience. Yeah.
My brother's a molecular cell biologist, and he had a big argument with his boss because they disagreed about a particular function of folic acid in digestion, and it became very bitter. So then he started up his own lab, and now he's his own boss, and he probably has the same arguments with the people that work for him now.
Well, that's maybe why I'm a little bit cynical. I hate to say it. Maybe I'm a little... Come sit down, come get involved with this. We're having a conversation about whether it's possible to have science without bosses. Without a boss.
What? Can you
have
science without a boss, Alona?
So I'll just recap very briefly, because, I'll just introduce the people around the table. We've got Mikey, we've got Abby, we've got Julia, and Ilona. It's very nice to meet you, Ilona. So Julia was just saying that one of the ways that we might have science without hierarchies or bosses would be if everybody had a basic level of education in all of the major fields that are required to do basic science, and the arts. We made that clear that was also very important, that people should have a certain amount of creativity and faith to nature in the way that they were doing observations. But then we also needed to have some form of respect for experience and knowledge, but that might be what we'd need in order to have all the tools, all of the basic resources in place to have science without bosses. And then Mikey told an anecdote that his brother had a quarrel with his boss once. But I did think that there was something to that, which is that ideas do have a certain kind of personality identification. Maybe there is a sort of dynamism to the ways that people get frustrated with their bosses and have to come up with new ideas. So that's where we're at so far. But I think
to Mikey's point, my undergraduate advisor told me that I would know when I was done with my PhD when I hated my PhD advisor. So I think that gaining a certain level of scientific maturity actually brings you to the point to have the level of disagreement that you can with a so-called boss.
Okay, so there you go. There's the second anecdote, but it's a personal one, not just about your brother or sister. There is a requirement for some form of antagonism, and maybe that means you also have to own your ideas a little bit if you're going to be antagonistic with others. Go ahead, Elena.
In my experience, this is true for both academia and the industry, the best boss is the one who doesn't have to make the name for themselves. They give you the best intellectual freedom to do or pursue the directions you want to go. And but it doesn't mean that they have to be senior in terms of age necessarily, but those who kind of fulfill their ambition and they do not micromanage.
Okay, so they have to be wise in their accomplishment of their own goals.
Right, so I don't mind bosses, but I do mind certain kinds of bosses who do not give you any space for intellectual freedom, which is the key in science.
Is it also that's
for innovation, at least?
Sorry, what was that?
Or at least for innovation.
Okay, so you can do science without these wonderful, selfless, wise bosses, but it won't be innovative. It might be something else. It might be is that the norm?
It's just going off the subject a little bit, but you know, one of the things that I hate most in life is a TV program called The Apprentice, because of what it kind of establishes as a norm of what a boss should be. It's kind of like hiring, firing, all-powerful kind of decision maker, rather than, in my mind, the apprentice should be
indispensable employees. That's what comes hand in hand.
It's my feeling though that, I mean, talking to people today around the table, it seems that there are lots of people here struggling to hire talented researchers who have a high level of methodological training, are ready to go into industry and start doing bioinformatics at a professional level. So they're not just...
They might not want to listen to what they have to say.
Well, I guess that you did say that this is in industry and in academia, but maybe there's some differences.
Yes. Well, I think fundamentally there are no differences. The way I work in different labs and different companies in the industry, and I find it's really the philosophy that divides, those who are seeking to control your every move, and those who believe that they have to hire smart people and give them resources and the freedom to basically do great work. You have to trust your people.
It's failure. Science includes failure, absolutely.
Yeah. So.
I have to say, I do love this picture of science that's developing from your response to the idea of science without bosses, Julie, which is that everybody's well educated. Bosses, from your perspective, Ilona, are wise, they're selfless, they're mentors, they're willing to embrace the innovations of their students, and that I'm wondering though whether this idea of antagonism, wanting to hate your predecessors, isn't that somehow also important? I think that that's so much a part of the way that science actually works.
Maybe in their formative training years, that's important. I think once you've matured and become part of the contributing force, it's sort of passé.
You let go of the hatred?
I think so. I think you just become collaborative and peer-like, and you distribute tasks according to skills and interest.
Yeah, go ahead.
I mean, yes, this question, I mean, I'm not a scientist at all, but one of the things about ideas is they do get attached to a sense of ego, a sense of identity. Personally, I don't think that's a good idea. I mean, the idea, it's not an idea, it's a good phenomenon to attach an idea to an ego, because then you're not willing really to develop that. You're kind of offended personally, on a personal level, if someone challenges that.
Well, one of the things that we do in science is that we do have double-blind peer review, which is exactly about trying to separate out some of the things that go along with the ego and some of the things that go along with the personalization of ideas from the peer community. I mean, but the fact is, I'm not sure that it really works that way. And although I do love this picture of the future of science as a non-hierarchical social activity, I don't necessarily believe that we're going in that direction. So I'd be curious to think about, like, should we extend double-blind peer review so that maybe the paper, once it's published, is also not attributed to a particular researcher? How are we actually going to get there? Because is this the world we live in now, or is it the world that we need to move towards? Do you think we live there now already? So then how do we get there? That's kind of my question.
I don't describe to say the
To Julia's idea?
No, to the idea that once something is published, it should be somehow detached from the source.
That was my own probably very stupid idea as to how do you move beyond the...
How do you take the ego out of science?
Right, okay, because that does sound to me like what you're prescribing, and maybe that is what's required. I mean, you yourself said that you needed to have a selfless, wise
personality,
advisor.
right? There are extremely egotistical Nobel Prize winners, and there are extremely humble Nobel Prize winners that if you didn't know their name and what they did, you wouldn't know that about them. Whereas another might be telling you that every fifth word he's saying. I've met both types. So who knows, right? The institution of science is archaic right now, right? We're not even with pace with the rest of society. So what are you going to do? You know, you have to train your kids well and make your home fun.
Okay, well that is one suggestion. I mean, I like that. That's a very nice suggestion. If you just raise...
Yeah. George, come and have a quick seat on the table. We're talking about ego in science.
We're also talking about how do we... That's okay. You've warmed up now.
Yeah, exactly.
We're also
now
talking
you're
about
here for the proper talk.
the future of science. How should we go about creating the science of the future that we want? The science that is oriented towards sharing great ideas, maybe, rather than progressing in our individual careers and standing on the face, or standing on the shoulders rather than the faces of our predecessors. If that's a good way to say it. Do you have any ideas about this?
Plenty of ideas, but I'm not sure I should share them.
Well, please do. That's exactly what Talkaoke is all about. Before you leave, Abby, we just want to get a final thought. Just wait for George to come up with his first, and then we'll have your last.
I mean, the way the system works, actually, there are biases built in the system, both in terms of funding, in terms of publications, and everything else. So we have to inherently kind of redefine the system if we want to do this large, science that involves multiple groups where not everybody's going to fight who's going to take credit and who's not. Not everybody's going to fight who's going to take a bigger piece of the pie, who's going to take the small piece of the pie, and that's a little difficult in this day and age.
Whole system rethinking. Yeah. So Abby, you strategy, but let's just have a final thought from you, because you've sat down and I think you appreciate what Julie had to say, but you didn't actually add much about how
I came up with the idea of mentoring, right?
That's true, you did. I'm sorry.
I feel like
I attribute that idea to you.
So I still think there's someone who needs to pull all the end together, and that will be a mentor. And it truly depends on the person. Like every scenario is different. I've worked with both kind of process, but I always see the good piece, and then it starts reflecting, and responsibility keeps going, and I think eventually everybody will do that. But I don't know.
Is ego good for science? Is ego good for science or bad for science?
Confidence is good. Ego is not good.
That's a very profound point. Difficult though to separate them out, I'd say. And I think
not at all, not at all.
Okay,
I don't think so. I mean, I think confidence comes mixed with obvious humility, where ego does not.
Okay, so you can have humility and confidence, but you can't have humility and egotism. They're sort of antonyms. Okay.
Yeah.
I wonder whether there's two different approaches that I'm hearing at the table. Come sit down, come get involved. This is a kind of talk show, it's Talkaoke. At the moment we're talking about whether it's possible to have non-hierarchical scientific progress, whether we need to reform the whole system and the practice of science as a social activity, or whether we can educate our children in such a way that they naturally get involved in science in a wise and selfless way. So if you've got something to say about that, which I'm sure you do, come sit down. It's just... Sorry, what are you saying?
That was a lot, but...
That's... Yeah, go on.
How big is your ego? And does it determine how well you're going to do in science? Do you need a big ego to get far in science? That's the question. Julia says no.
Well, actually, hang on. Go ahead, Julia.
So I think I think in order to continue the conversation, you have to make a distinct. So we started out talking about, can there be non-hierarchical science?
Yeah.
The current state of science is absolutely hierarchical. There isn't anything less hierarchical than the state of science today, right? So now we're having a totally different conversation, which is, what about the hierarchy of science and how do you deal with that? So can you then navigate the hierarchy of science and the requisite for egotism to get to the top there with being a citizen scientist?
Well, that's a slightly different, that's also a new idea, citizen scientist. Is that a role? Which position would that have in the list of authors? Say you have a citizen scientist, are they going to be like towards the front of the paper, somewhere in the middle of the paper?
Physics, they do it alphabetical now. They're over it. Us biologists are way behind.
Okay, so biologists had that list of authors is like a ladder.
I thought we were done with this when the BRCA paper came out. You know, that was 20 years ago, over 20 years ago now, right? So come on, 100 authors on that paper. Everybody was supposed to have a piece of it, but who does? The first and the last. That's it.
Those are the big ones.
Well, having originated from physics, so my PhD was actually in physics, it is definitely a different world than in biology, and how the information is actually conveyed, both in the public preprint archives, so publishing the paper and making it available online prior to publication, and also not chasing the prestigious journals. You publish in the journal that is actually the topical journal of what you're doing, rather than the journal with the highest impact factor. So it's a little different approach. So,
a bit less hierarchical, perhaps.
Well, the hierarchy is pervasive. It's the journals, it's the authors.
Yeah, there's still a hierarchy in physics, but because it's academic primarily, right? But beyond that, I'm not a physicist, so I don't know for sure, but my physicist friends tell me things are much more egalitarian in that world.
Well, that's what I've heard as well, because I think they have a reputation for sort of year zero thinking that anyone can come up with an idea that would revolutionise the field, and they're quite open to that. Is that not true? That's the reputation.
See, now I'm not in physics, so I can come back and say. Physics is a very established science in comparison to what we're talking about here in genomics, right? We're talking about very well-defined theory, very well-defined experiments that actually correspond to the theory and pretty much in agreement. When we're talking accuracy in physics, we're talking like the 8th digit of a number. Versus when we're talking accuracy, I was like happy, we get 70% correlation, like yay, we're doing good. So it's a different discipline altogether. So genomics hasn't matured to that level of the science yet, and hopefully it will. On the other hand, it involves people, it involves a lot of industry that is not actually involved when we're talking about things like physics. Stakeholders are different. Let's put it that way.
I mean, I'll share a small anecdote in the middle, so I'm not really meant to do that. But my boss is an ex-physicist, and he is both aware of hierarchies, but also willing, because the theories that he works with are so well defined, so exacting. If somebody came up with an observation that definitely challenged something that he believed in strongly, he would have to accept it and move on. Whereas I think where you have less clear...
have to verify that it wasn't a mistake.
Right. Given that, I get the sense there is, in physics, at least the commitment that if you verify that you've actually found something, that observed something, and it falsifies some part of a theory that needs revision, people will do the revision. Whereas if you're dealing with 70% correlations, maybe there's more of an appeal to authority as a way of establishing whether or not you do need to revise some basic and maybe uncomfortable structural facts about the way that the field's put together.
I just think it's really loose. Even in the case of an agreed upon retraction, it can happen that that retraction never actually occurs. So the scientific community is actually never told about that, and there's conflicting information in the field. Now, it's minor, and so a lot of science that's done in the world is not interesting to everybody else, and so you just won't ever find these conflicts in the body of science unless you happen to be the one that, you know, produced the data that put the other data in conflict.
Right, or you read retraction watch.
Or you happen to do that. Who does that?
Well, I think there is now. I mean, certainly in some fields, there's a real effort to kind of reform some of these institutional structures, and maybe that's a good thing. Some people also are calling it, in psychology at least, there's this methodological terrorism accusation. People heard about that. There are people who are more or less interested in reforming these kinds of structures, because obviously there are people who have interests on both sides. So I'm quite intrigued by the idea of how we move towards this. I got the sense earlier from what you were saying, Julie, that actually these are more about personal values and the things that you learn in a home and in a school and in a university. As you educate people right and you mentor them correctly, that was Abby's idea, that maybe they'll just make better scientists. Whereas I got the sense when you said that, George, you mentioned that there are also whole systems of established practice that need reform. So how do you move forward with that? I mean, this is Talkaoke, it's a talk show, but sometimes people have great ideas here, and sometimes those ideas go somewhere. So maybe we're on the threshold of one of those moments. If people come up with some ideas about how to move this forward, then I mean, that would be a real accomplishment for 3, 4, 5 on a Tuesday afternoon.
Very idea.
Go ahead.
It goes back to the conversation about confidence, egotism, and humility. I would love it if it were awesome to be an anonymous, ordinary, unglamorous scientist who never won a Nobel Prize and no one knows your name. You know, like, there's tons of us, especially when you come to the whole conversation about getting children and girls into STEM. Well, then it needs to be okay to be, like, boring and ordinary. Okay. You know, and just like the ordinary the ordinary endeavor of doing science is a really good contribution to make. And you don't have to be a Nobel Prize winning scientist to be a great scientist. You can be a totally anonymous scientist to be a great scientist.
Did you have something to say about that?
Yes, well, just to be realistic, there are practicalities in life. I think this is a way too far removed from this idea yet. But I think the concept of
come get involved in this.
Of citizen science, I think brings us closer, because there are plenty of people who don't have formal education in biology or physics, but still wants to participate in what's happening in science. And I think they keep asking questions, legitimate questions. And I think it brings us closer to this concept of democratization.
Okay, so I mean, those are two quite different ideas, but they're kind of married together. One is that maybe you could have people outside of the institutional realm of science who can contribute. On the other hand, you could make the institutional realm of science less dependent on celebrity and a kind of unifocal life that is devoted entirely to science in some one-kind way. Michael, what do you think?
No, I just think, you know, what's the definition of success? And I think this is, you know, the celebrity culture is where we're at. So we're kind of going more in that way and less about.
Come sit down. This is Talkaoke. That's good.
And it's, you know, it's less about, you know, doing a job and contributing to the field in an unglamorous and anonymous way.
Okay, so welcome to Talkaoke. I'm just going to go around the table quickly and introduce you. This is Mikey.
Mikey, we've got 10 minutes.
Okay, we've got 10 minutes. This is George. What's your name?
My name's Mary.
This is Julia and Ilona. So far what we've been talking about, when we started talking about, is it possible to have non-hierarchical scientific development, or the knowledge of the world, some of the world's knowledge about science could be moved forward without the kinds of hierarchies that we see in institutional structures, in authorship structures, in incentive structures of scientific career. We've just been brainstorming a little bit about how can we move forward with a more, maybe a utopian vision of the future of science. Julia suggested that we could have less of an emphasis on celebrity and the focus entirely being on career achievements. I think there was also a sort of an issue of the sort of gendered norms of science, and that maybe it's okay to also have a family and concentrate on things that are not your great idea. Ilona then brought in the idea that maybe citizen scientists could be appreciated and involved in the development of science from outside those institutional groups. So I don't know if you had something to add on that subject, George, but that's kind of what we're talking about. This is Talkaoke, so we can go in lots of different directions. I saw you nodding when I said citizen science, which was Ilona's contribution. Did you have something?
Yeah. So what I might say is you are also capitalizing on the hacker mentality that we see so much of in Silicon Valley / Boston / in other countries. So, and I think as a counterbalance to all these large scientific initiatives where you have 100 people on the papers, that also takes other ambitious people that maybe aren't on those projects, or even people on those projects who want to make a name for themselves, might hack the system through the citizen science projects.
Okay, so we're talking about kind of open source citizen science, or hackers. I hesitate to use the word hacker because I think that's got a negative connotation, and maybe it's even a bit scary if I think it would. No?
I disagree. I think the hacker is a very good word, because you're kind of, it kind of calls upon that sort of idea that you're going to reconstruct and reinvent stuff, and just DIY it a little bit.
Yeah, maybe DIY. Yeah, DIY. So I think hacking within the ethics, the scientific ethics. So maybe democratizing it a bit, but still staying within the tradition.
Okay, I have to say I'm liking these ideas. You're looking a little bit skeptical, Julia.
I guess I don't know enough about hackers to know how well they know their field, but I guess I don't know how somebody coming in from nowhere would know enough about regulations of the space.
So there's actually
Should be well.
in the Bay Area, and I think they also have a lab here in Boston. Indie Biolabs is actually considered a biohacker space, and they just surpassed. They are the largest fund if you count them by number of startups created. So I do think there is a biohacker movement, and in the Bay Area, which is where I'm from, these people are actually they're right down the block from, I'm trying to think of they're right down the block from Berkeley, sort of across the bridge from UCSF. You don't necessarily get the Stanford people crossing the bridge, but you've got a huge cluster of really skilled synthetic, I think it's more synthetic biology, more than it is traditional.
Okay, so maybe part of the future that you're imagining is already here. There aren't enough people maybe crossing the bridge yet. That sounds like quite a good metaphor. But,
Yeah, I mean, I'm not a scientist, but I'm probably more of an anarchist than most people. And I just think it's just a better way. It's not necessarily about doing better science, but just a better way to structure things for people.
I also think you see it in Y Combinator is really strong outside of Stanford. So the Stanford people go to Y Combinator, but she was asking who funds it, right? So Y Combinator is actually funded by PayPal money, and Indie Biolabs initially was funded through Google money and PayPal money, and these people get micro grants, and they get about 6 weeks to 3 months to do proof of concept biology.
So So this is the kind of hothouse model from Silicon Valley applied to biology and biotech. I mean, is that where we should be looking for this non-hierarchical future of scientific development?
Right at the very beginning, we were talking about a different subject, but it fits in, which is like how to get people more, like a wider group of people more interested in genomics, and how to actually make sense, get people to understand that makes sense to general population. And one of the things that was said was this kind of idea of a kind of garage genomics.
Yeah.
And you know, this is actually effectively what seems to be happening here, and I think it's a really good way to actually make it more exciting, more sexy, more glamorous, but also, you know, more embedded into culture at large.
I have to go to Julie here because she's making some real disgust sounds.
Yeah, well, I mean, so I feel like we're already in, okay, so like, I'm gonna get killed for saying this, but we're already in the world of garage genomics, right? There's no standards, there's no cross-referencing, there's no, I mean, it's really like Wild West. It's crazy. And so, to take it even more, what do we want to do? Go like 100,000 years in the past and become Neanderthal genomics? Or like, I don't know what we want to do. Genomics has already diverged far enough away from traditional genetics and the basic understanding of how this stuff works, and hasn't made sense of so many things that we've now observed. And like, everything we've known has shown us that our pre-predictions to this knowledge were wrong. Like before the whole genome was sequenced, every expert in the world was saying 100,000 genes. We've got 100,000 genes. Okay, we have 20,000 genes after counting. So, and that's probably wrong because of our whole concept of gene is going to get totally revolutionized. So, garage genomics, I don't know. Maybe if like the people in the garage are like the Steve Jobs character type people who fully understand like real actual genetics and all the weirdness that can happen there, and that involves some really heavy stuff. So like the stuff that we have our hands on right now is nowhere near where it needs to be to have a real impact on human health and understanding of genetics in general.
Okay, so I get a sense there is actually, although you have been advocating for the democratization to some extent of science, or the spread of the kinds of involvement that people have in science as a career path, there's also a sense of respect for science as an endeavour that is not just about involvement, but is also about knowledge, and is also about a certain kind of knowledge and a certain respect for that knowledge. And I wonder if there may be also a bit of a contradiction then in trying to change this from a field which does have lots of people accessing it. Maybe it's garage. Maybe the argument actually here is between, do we want the kind of Steve Jobs garage genomics, or do we want the kind of Mad Max garage genomics? It depends which one is
Genomics.
okay.
Well, in defense of Indie Biolabs and Y Combinator, they actually have scientific advisory boards, and their applications are vetted through a scientific advisory board. So there is an application process. It's not just any Joe and E. coli showing up with a couple of CRISPRs. Let's make potatoes.
Okay, but then I have a question for you, just because at the very beginning of this discussion, you said actually what we need to. Okay, well let's just get. I think we're going to wrap up here actually, because this is the end of the day. We've got our drink vouchers too.
Oh yeah.
To cash in before that opportunity passes us by. So we're going to have some final thoughts to wrap up this conversation, and in fact the whole first day of the conference. So I was going to start with you, George. If you guys are going to run off right now, I can start on this side of the table. But if you could, are you happy with this? Okay, we're going to go around in this direction. Mikey, I'll come to you last, because you're in fact, this is where it starts. You said at the very beginning of this conversation, George, you thought that the whole infrastructure of scientific knowledge development, as it stands, which is very linked with these arcane academic structures, needed reform from the top down in a way. Now what we've heard is a lot of ideas and counter suggestions about how that might happen from the bottom up. What's your perspective on a realistic route forward, or just generally final thoughts?
Probably more less severe way forward. I mean, let's do simple things. So anonymous evaluations of research. All we need to do is be able to evaluate research objectively as scientists, right? So I would say anonymous evaluation of papers, anonymous evaluations of grants, anonymous evaluation evolution of science in general. And that's the first step. I think that would solve a lot of the things that we're discussing, including the ego question.
Okay, so we can bypass the ego through anonymity. It's been tried online. Was it a good idea? Or what's your final thoughts for the day?
Oh, well, just to build off your thoughts, I think there's pure science and genetics. There's commercial science, and then there's also new applications that are developed. And I do believe that for new applications that are being developed and early commercial science, since we've seen a decline of NIH funding and an increase in private funding, either through wealthy individuals like we see in the Bay Area or foundations like we see coming out of Seattle, there is an opportunity for us to take these talented scientists that maybe are on these huge papers of 150 people and let them have a small proof of concept funding to explore a new avenue of science to build tools or products for the rest of us.
Okay, thank you very much. That's a very thoughtful, well-considered, rounded final thought. Julia, you kind of kicked a lot of these ideas up into the air. What do you feel like rounding off with?
I think the only thing I really want to say is, so if we look at the proliferation of genomics, especially, commercial enterprises doing something genomics related, we could just imagine what that field might look like if everybody involved got out of high school with basic biology, chemistry, physics, and math. Let's just say that. It's like we don't support that with our American basic public education. As a high school student, you get to choose one of those and call yourself done. So I think you're not done as an individual unless you've gotten the basics of those in addition to all the other things we've kept in the curriculum.
Okay, so I think that's a good another appeal for education, which is kind of where we started. I feel like it's now kind of made a little bit more coherent by adding these other ideas about anonymity and removing the ego, maybe proliferating the kinds of spaces in which it's possible to do science and separating them out, and also having a more generally educated population. Ilona, you all got to be, apart from making the final thought for the day, so what would you like to add to that mix?
I think what I'm seeing outside is that technology companies are moving into the life science space, and I'm kind of excited to see it, but at the same time I think it changes the landscape. The expectations are different in the technology space because things tend to change and happen faster, and this is not applicable in life sciences most of the time, unfortunately. So I would like to see a better understanding from the technology companies' side of this reality, and once I think the understanding is reached, we will be better off to move this field forward, because you cannot. Right. But I do believe that this is an essential step to bring this field of genomics to the next level, where we can actually capitalize on it in an unimaginable way.
Okay, that's a wonderful final thought. Thank you very much. Mikey, did you have a final thought to add?
Yeah, from my interest is like, there's a lot of science here going on, and a very, very fast-paced development, but how much science is being done about how the science is done? Do you know what I mean? No, people don't really test different models of hierarchy or different models of approach and actually compare, or I don't know.
Julie,
it's like,
she's just disagreeing with you, so I just want to ask you to...
They do if they take a systematic approach, and it's true that in biology, I think it's primarily dollars that limit the ability to take a truly systematic approach. But when you're lucky enough to be in a situation where the dollars support that, you can interrogate your questions systematically and build the matrix correctly.
So sorry, Mikey.
Nothing more to say from that, apart from let's go get a beer.
Okay, well that's a great set of final thoughts. Thank you to everybody around the table. This has been Talkaoke. We're going to be here tomorrow all day.